Good afternoon, children. Now we were in our nervous system. If you remember, neural control and coordination we finished, and the end of the lesson is actually sense organs. So today we'll take only the sense organ alone, and we'll proceed along with that. These sense organs, you know very well. You have to sense, that is in our neural control and coordination. Why do we come to the sense organ? You know very well, sorry. Then the entire nervous system is based on stimuli and response. Okay, stimuli is anything which makes you react, and the reaction is the response. So this excitability or receiving sensation and answering, this excitability and capacity is the major conducting capacity is the major function of the nervous system. So when we studied the different parts of the central nervous system, peripheral nervous system, that's completely involved in the nerves. And CNS, we studied about the structure of the brain, main CNS, the brain, different parts of the brain, and the functions of the different area. And now we go to the sense organs. The sense organs, as it is, if you take about. So what are we going to do in sense organ? Now the sense organ is first in your body. Sense organs are organs which receive the stimuli. They receive stimuli and pass it on to CNS, central nervous system. From where the response is given. Response is brought by the motor fibers to the effectors. So in another word, the sense organs can be known as the affectors, and the motor area organs are known as the effector. This is what we are studying. The peripheral nerve system, the nerves have an effector fiber, sensory fiber and motor fiber, or which is effector fiber and affector fiber. Effector is effect, action is produced. So if the action is produced, that's a motor. And affect is which is producing the action is the affector. So sense organs, or in another word, can be the sense organs receive the stimuli and conduct the stimuli towards the center where the stimuli is received, analyzed, interpreted. Answer is immediately sent by the region to the effector organ. So every stimuli will have the response, that is an action. So affector and effector. Sense organ or motor organ. Sense organs are the ones which receive the stimuli. Motor organs are the ones which respond to the stimuli. So this is a basic principle of the entire nervous system. If we don't receive the sensation also, our nervous system is not functioning. And if we don't respond also, our nervous system is not functioning. All this part we saw in the central nervous system, how the motor, where is the motor area, where is the sensory area. In your main center is your CNS, the brain proper, cerebral cortex. So all the sensation from the entire part of your body, it practically goes through via the sensory fibers to the sensory area and the cerebral cortex, where it is received, analyzed, interpreted, responded. They respond to the next, you know very well, cerebral cortex has got motor area and sensory area side by side. So the response is given by the motor area, and all the axons from the motor area form the motor fiber or motor nerve, which comes to the concerned effector organ. Suppose I have a pain. So I'm poking, something I'm poking, it's not a poking, a cut. You know very well, you are cut and the pain is there. And immediate response, what should you do? You press the thing or stop the bleeding or put the bandage. All these responses are voluntary. You are aware of your cut, you are aware of your pain. These are the sense organs when you study about. You are not talking about the reflex actions. Now you are talking about these organs which helps you to receive the stimuli and send it to the central nervous system and get the response. Inner organs. This is normally these sense organs are on your external surface of your body. The inner organ, viscera. Now the viscera also has got sensation, feeling of hunger, thirst, temper, anger. So all these are the inner viscera. Suppose your secretions, stimulation of the to produce a hormonal secretion or gastric secretion. So the reactions of sensations and respond of the visceral organ is completely under the autonomous nervous system. See, this is completely when you talk about the sense organs which are on the peripheral part of it is under central nervous system. But if you take about the sensations or feelings or visceral sensations or the sensation of the separate organ, what do you mean by that? Now, you know very well, we studied the gastric secretion. As soon as the saliva secretes or the salivary secretion stimulate the gastric secretion or mastication of the food, smell of the food increases appetite. So these sensations are the visceral organs. Or when you study in the nephric filtrate, if you remember, blood vessels, blood pressure, alteration of the blood pressure, increasing and decreasing the heartbeat, increasing and decreasing the filtration rate. All these are the functions of the inner organs are under the control of autonomous nervous system, which has got sympathetic and parasympathetic fiber, which are antagonistic to each other. But here when we talk about the sense organs, we are talking about the major senses. Senses, what are the senses and what are the sense organs related to that? Okay, so you will be completely studying about the different sensations of, we will sense organs of eye. Extrasensory perception is different. That is, there are people who can feel extrasensory perception is beyond your sense organ in your body. That's the sixth sense they talk about. You know very well there are major 5 senses. They namely touch, sensation of touch, sight. So what are the sensations? Now talking about sensations and sense organ. Sensation of touch, then sight, then audition or hearing, then vision or sight, and taste. And the concerned sense organs are sight we have written already, vision or sight. Now touch, smell, electrotouch smell, and smell and taste. Smell or olfaction or sense of smell. Then taste. Now you know very well the sense organs which are related to that. Touch is mainly skin, and sight is your eye, and audition ear, and olfaction nasal epithelium. And taste is your tongue. Now these are the organs. Now these organs have a particular region which will receive the sensation. Now those parts are they are nothing but the nerve endings, nerve fiber. You know very well neuron, the fibers of the neuron forms the nerve, the axon of the neuron forms the nerve fiber, bundle of nerve fiber forms a nerve. The axon, you should not imagine one axon from the brain is extending up to the end of your toe. They convey the relay, that's what you mean by the synapses. And the nerve, when a bundle of nerve fiber comes, it becomes a nerve, and the nerve completely ends in an organ. When it ends in an organ, it will definitely end up separate into the axon, and this and all we saw as a motor end plate. Similarly, the end of the nerve ending or the axon of the nerve fiber will, when it reaches a sensory organ, a particular area of the sensory organ, it is a synaptic nodule. Normally, when a nerve axon ends on a motor organ, muscle or a bone, it will end in a motor end plate. When it ends on a sense organ, it will be invariably synaptic knob. Alright, now this part, the conduction of the nerve impulse from the motor end plate to the muscle or the synaptic knob to the particular region there, that is the transmission by the chemical acetylcholine, adrenaline, the motor end plate and the cleft, and we saw this previously. If you haven't seen, you have to post it to them if you are interested in that. Now listen, now these sense organs have got the particular area where the sensation are felt. Now the touch sensation is a stimuli. it should be received. So that area where it is received are called sensory receptors. They are called sensory receptors. Sensory. These are the organs, related organs. Now the particular area, they have sensory receptors. The receptors, in a sense, it can be a simple nerve ending and an axon ending branching into the foot plate. Or it can be that, definitely it will be a nerve ending axon only. But it can be modified in a form of a structure. Okay, now for instance, what I mean by that, when you talk about the eye, you know very well rods and cones, there are sensory cells. But the axons are the optic nerve, is the ganglion of the optic nerve. Only the main receptor conducting, finally the stimuli is received by the nerve, optic nerve. Optic nerve takes the stimulation to the brain. So every organ has got a sensory receptors. Now the skin has got various sensation, it has got various receptors. Now what are the receptors in this eye? The sensory receptors are, we call them as the rods and cones. Rods and cones. Now you have different receptors. This we will see the different receptors. What are the sensations? Each sensation has got different receptors. Skin receptors. Now if you take there are two sensations here. One is balancing, equilibrium, another one is hearing. So mainly you have got the ear, inner ear. That is, in other words, you have got the organ of Corti, that is for hearing, and structures called cristae and ampullae that helps in maintaining the equilibrium. Whereas this cristae and ampullae, they are connected to the semicircular canal. So the end of the semicircular canal and vestibule, you have got the inner ear structure of the inner ear. I'll explain in detail if you have the time today. And otherwise, we'll take it in the next class. There, if you study, you have got special sensory receptors in the ear. There are three types of receptors. One is organ of Corti in the cochlea. That helps in hearing. Now, vestibule, utriculus and sacculus. In utriculus, you have got a sensory area that is called maculae. And ampulla means end of the semicircular canal, swollen end, the ampulla has got cristae. So cristae, maculae, organ of Corti. All the three are in the ear. One is for hearing, that is organ of Corti. The other two are for balancing, movement in a acceleration of the movement, in the slow acceleration or horizontal or vertical, different acceleration, maintaining the equilibrium of the body. When the acceleration means movement of the body. Now the movement can be up and down or downward, forward, horizontal movement or circular movement. So all these movements or accelerations or movements of your body are controlled by the other two receptors, that is called organ of Corti and maculae. Now where are they? They are connected in the inner ear only, connected to the utriculus part, that is in your semicircular canal. Next, when you come to the nasal epithelium, epithelial lining. But the epithelial lining have got special cells which receive that sensations. And taste, tongue, you know very well, taste bud. Taste buds are in the tongue, and the different types of taste buds, which the first lesson you should have studied, and what are the different sensations. So now we will take up these things which are smaller, and then skin, eye, and skin, taste and smell. Then we'll come to the eye. Last we'll go to the ear. Alright. Now these are sensations. These are sense organs. Every organ has got a particular area where the sensation is received. And when the sensation is received, you should know from these organs, the sensation is carried over to the CNS. So from there, you have got the fiber, the sensory fiber, organ of Corti, and rods and bones. Now skin, you have got the nasal epithelium, taste bud. All these are forwarded to your CNS, cerebral cortex. Where now, for example, you take eye, optic nerve, ear, auditory nerve. You take the smell, olfactory nerve, with the first nerve. If you take here, tongue has got two supply. Sensory nerve is separate, motor nerve. What do you mean by that? Muscle of the tongue is supplied by one cranial nerve. The sensory receptors of the tongue is supplied by another cranial nerve. That the ninth cranial nerve takes the sensation. There is a glossopharyngeal. That's a sensory nerve. But the motor fiber which supplies the muscle of the tongue is 12th cranial nerve, that is hypoglossal nerve. So in this case, 9th cranial nerve. Now skin, the nervous ending completely reaches through wire from the skin. It is carried over to your receptors in your particular area. Where if it is in the toe region, skin is overall your body. So these profuse, that the skin is the major sense organ which helps you to maintain different sensations. These are particularly oriented in only one sight: hearing, smell, taste. But if you take the skin, because you are exposed, your entire skin is exposed to the environment. So it has to face various stimuli like pain, pressure, temperature. So these are the major sensations. So first we go into the skin part. This is the gist of the lesson. So as soon as we finish that, we'll follow all these organs. All right. Now we take the skin. First we take the skin. If we take skin, the sensations are pain. I'll go to the pressure first. Pressure, pain, and temperature. Now, in this temperature, you have got cold and warm. Hot, we won't say hot, cold, and hot. Now, in the case of pressure, you have got light pressure, very light pressure. In a sense, suppose you are walking on the road and there is a heavy breeze. Now, the atmospheric pressure is around you. That's another question. Your whole body is under the atmosphere. Atmospheric pressure has got a value that is 760 mm Hg. Okay. Now, but you don't feel the atmospheric pressure one bar pressing your body. Why? The reason is you have got the respiratory system, you have got the residual volume, the inner organ, we have internal pressure, that is the body fluid, and the lung filled with the residual volume. So the body fluid as well as the intracellular fluid, lymph, and the blood vessels, the blood in the blood vessels, as well as the residual volume in your respiratory tract. That's why the lung does not collapse. Completely, the lung is emptied of no air, it will be just collapsed lung, it will become. So these structures, particularly inner your body, counteract the atmospheric pressure. That is the main reason you don't feel the atmospheric pressure pressing you. Though we are surrounded under certain volume from atmospheric pressure. Now, and this is another reason when you breathe in, the volume of your thoracic cavity increases, the pressure inside the thoracic cavity decreases, or the lung decreases, so the air enters in. Reverse occurs when you expire. So now when you talk about the pressure, light pressure, suppose the air is blowing on you, you can feel the thing on the hair moving on. Light pressure and very heavy pressure. Where somebody is pressing or carry a load, the heavy pressure. And in this, now you have got, there are many receptors. The pressure receptors are known as Messiner's receptors. Now, these are the sensations, okay? Pain, temperature. Now, what are the receptors in the brain? The pressure is actually mechanical. Pain and pressure will come under the mechanical stimuli. When you talk about the, now this is regarding the skin alone. All are mechanical stimuli only. Okay. Now, these mechanical stimuli, now, when you talk about the pressure receptors are known as Messiner's receptors. Now, what are the receptors of these pain, pressure, and temperature? Now, now the receptors of the pressure is known as tango receptors. Tango receptors, phonoreceptor, stator receptors. Tango receptors are the one which helps you to feel. All these three are the receptors, okay? Messiner's corpuscle, Merkel disc, basket nerve ending. You can mark it down if you are interested. Now, this tango receptors particularly helps in the light pain. Now, phonoreceptor, pain receptor, pressure receptor, slight pressure. Now, the phonoreceptors are Puccinian corpuscles. Puccinian corpuscle helps to feel the high pressure, heart pressure. These are for light pressure. This is a little bit more pressure is heavier pressure. Now, statoreceptors helps you to maintain your equilibrium. That is when you in the movement in the acceleration of your body. That is when you turn the head or your body movement. Now that is statoreceptor. Under statoreceptors, you have got what are the receptors, or particularly the inner ear, and all the three receptors of organ of Corte and cristae and macullae. Now this is the inner ear, organ of Corte, cristae is in the ampulla, structure of cristae, then macula is in the maculae. Macula is in the all these two are in the cochlear region, organ of Corte. These two are in the vestibular region. Now this is in the cochlea. This is sense of hearing, and these two are like movement, the acceleration of the body in different angle. Acceleration is straight line. Now mainly these are organ of balancing. What do you mean by the organ of balancing? Now your movement can be forward movement, upward movement, lateral movement, or circular movement. Now all these movements your body should be able to maintain the equilibrium. Now that is the organ of equilibrium. So now these are all the pressure receptors. Now these mechanism, how does the mechanism? What is these receptors? They are nothing but nerve endings. They're axonal ending. But the end of the axon will not be a free end that could be surrounded by the cell membrane. It's only a continuation of the cell. It will be surrounded by a membrane, a little bit of cytoplasm, and it's granular. It has got a particular structure. Now, if you take the Messinus corpuscle, the Messinus corpuscle, the axon will end up like this, and it will be like this is the axon. If you take, it has got a circular structure like this way, and the tip of the axon is a number of fibers will be surrounded. Cytoplasm, there will be a number of granules. This is Messinus corpuscle. Now, this is called the why the word comes funny. This disc, this one will be, suppose this is an axon, and the branches ends in a number of branches. Every branch will have a disc-like structure. That's why it is called Merkel's disc. If you take basket nerve ending, it's mainly present in the, now this is tango receptors connected to the skin. So all these are in the, these nerve endings will be completely ending in the epidermis. Now first you should know the structure of skin. What is the structure of skin? Skin has got upper part epidermis, lower part dermis. Epidermis has got the epithelial layer, 4 layers of epithelium. Epithelial layer, the topmost layer is a stratified epithelium, squamous epithelium. It's a dead tissue. It can peel off. Dermis is the lower part, if you remember. Now, how the nerves are in the skin, if you don't know, have a basic idea of the skin, you won't follow how it's ending. Now, if the skin has got two parts, upper part is the epidermis of the skin, and you have the lower part is called the dermis. This is completely a connective tissue, and the blood vessel, the fat deposition, subcutaneous fat. This is fat lobules. Now, this is dermis. In the dermis, you have got the innermost one of the epidermis will be folded. Now, epidermis has got 4 epithelial layer, complete lining layers of the epithelium. 4 layers. Innermost layer of the epithelium will be folded into called the malpighian, the papillae. Now, these papillae has got the malpighian granules, and that gives the color for your skin, pigmentation of the skin, and all these are the dermis. The nerves will end in the papillae. The nerve ending. So automatically, the nerves which are present in the dermis will reach the epidermal region because your sensation is on the upper surface only. You're feeling the receiving the stimuli. So the nerve endings only are known as these different nerves. They are nothing but end of a nerve. What is it? Axon. It gets a different shape. That's all. But all these nerve endings will be ramifying or branching in the epidermal cell only. Now, if you take the pains, the next pressure. Now, next sensation of the skin is pain is nothing but simple nerve ending. Now, here the nerve ending has got a particular structure. This is why it's called the Bastille Disk. Now, this one, what happened? This mainly present in the root hair. If you see the skin structure of the skin, now this is a dermis. You have the root hair. The base of the root, shaft of the root comes. It is surrounded by the covering and just got the sebaceous glands. This is a hair follicle. root of the hair follicle, shaft of the hair follicle, which passes the epidermis, which comes out. That is the hair. Now these fibers, the basket nerve endings, what will happen? The dermis, the nerve ending in the dermis will form a network around the hair follicle. Now this is the reason when you pull the hair, you feel the pain. Sometimes a single hair gets caught. It's very painful than pulling the entire hair. Now, so the stimulation which you receive, the thing is mainly the pressure which is applied onto you when you comb your hair, pressing it down. So these are the basket layer nerve endings. The nerve endings on the dermis, instead of thing, they will be forming a network around the hair follicle. Now that is the peculiarity of the. Now this is around the hair follicle. Around hair follicle. All these are on the skin. We are talking about the skin only. So now these nerve, they are, receptors are nothing but nerve ending. Nerve ending is called axon. Now these axons, end of the axons will get a particular structure. So the particular structure has been given a name. So these particular nerve ending will help you to feel the different sensation, receive the sensation. Your skin is the outermost part. So what are the stimuli which can attack your skin? Pain, pressure, temperature. So the pressure is a major one because with the unawareingly or awareingly, somebody pushes you or you carry a heavy load or a gentle breeze passes through or without your awareness, you are under the pressure of atmospheric pressure. So the pressure can be a normal pressure. You don't even feel every time you don't feel the air is pressing you. Now when you carry, you can feel the heavy load on you or somebody is poking you, move, pushing you. You can feel the pressure on you. So there are pressure in a different degree. It can be a lightest one, light one, or a heavy pressure. So these, according to the intensity of the pressure, there are different nerve endings receive the stimuli. That's all. So the engineer is very, very clever to make your body so intricate. So even simply one nerve ending and feel whether the pressure is heavy or light or anything. Why? Your response will be according to that. If somebody keeps on pushing, don't push me. You say, don't press me down. So, but when you are walking on the road, you won't tell the air, you don't press on my body. So you are not aware of the atmospheric pressure is acting on your skin. So the pressure are the different degrees are there. So according to that, the different nerve endings receive the stimuli, and this stimuli these are the nerve receptors. So the tango receptors are the one which receives the pressure. Now, in the light pressure, but in the tango receptors, normally in your book, but messiness receptors will be definitely given. Now, they will simply give messiness corpuscle. This corpuscle is different from the blood corpuscles. Red corpuscles, white corpuscles, blood cells also known as the corpuscles. But these are called touch corpuscles. Now, touch corpuscles general word. But this is the messiness corpuscle is a tango receptors, are receptors which help increase the pressure. Then another one is called phonoreceptor. Phonoreceptors are Puxinian corpuscles. Now they help you to feel the intense pressure, heavy pressure. This is light pressure, this is heavy pressure. Now Puxinian corpuscles, the structure of the Puxinian corpuscle, it will look like this. Puxinian corpuscle will have the nerve ending and like your the rays on your, you see the lines on your thumb, one on the other, the lines on your thumb. This is a peculiar funny one. It has got a number. This is the Puxinian corpuscles. Now this Puxinian corpuscles helps you to feel the heavy pressure. Stator receptors are the ones which are related to this. Now these are all. Why did I say the stator receptors are not connected to the skin? But why we call that as a stator receptor? All these receptors, whether tango receptor or phonoreceptor or stator, they are called mechanical receptors, mechanical stimuli, which is mechanically affecting you. Okay. Now the stimuli is mechanical. It can be chemical or it can be photochemicals. Now sunlight or electrical or electromagnetic. So the stimuli, different kinds of stimuli, mechanical stimuli. Mechanical stimuli, these are the different types of mainly mechanical stimuli acts on your skin. The mechanical stimuli can be classified like this way, and these are the organs. Mainly skin is the organ. So when you talk about the skin, next one is the pain. Now pain is received by RGR receptor or proprioceptor. Receptor and proprio combined together, proprioceptor. This is pain. You know, these receptors are simple. Now the receptor structures are a simple nerve ending. Now these structures will appear. So this is sorry. Now this is the epidermis of the skin. You have got a number of cells, epidermal cells. The innermost layer is thrown into papillae, epidermis of the skin. You have got a number of epidermal cells, 3-4 layers. You should know the structure of the skin in this. Outermost layer, staphyloid squamous epithelium, and stratum spinosum, stratum germinosum, spinosum. Now you have got a single layer of Malpighian layer filled with the Malpighian pigment, which gives you the sensation. These elevations are called papillae. Now these are the first layer, second layer, third layer, fourth layer. Fourth layer is the Malpighian layer, pigmented layer, and that is uneven thrown into fold. Okay, this is called papillae. Now these pain receptors, what you call that is Algiser receptor proprioceptor. If you don't want to know the name, at least you should know pain receptors. They are nothing but nerve endings. Now they are known as nerve ending. No name is given. The nerve ending is the term given for them. Suppose a nerve is there, what will happen? The nerve ending, it will branch out into a number of branches into the epidermal cell. There is a nerve ending here. Where is nerve? Nerve in the lower part, dermis of the skin. Now this portion is a dermis. The nerve endings are supplying the skin divided into a number of branches. These axons end over. They finally ramify or form a network on the upper layer epidermis. So the pain receptors are only nerve endings. Alright, now temperature. The temperature you have got warm. Sorry. The cold and warmth. Now there are two receptors to receive this is pain, pressure, temperature. Now the third set is temperature. Now this third set is temperature, this is pain, this entire thing is pressure. Pressure is mechanoreceptor, so mechanical stimuli, so we include the statoreceptors under this. But actually statoreceptors are not connected with the pressure. We can say air pressure because statoreceptors organ of cortex is a sound wave. Pressure is a sound wave only. That they respond to the sound wave vibration. So we can say pressure of the sound exerted by that. So statoreceptors come under the mechanical stimuli. So these are pressure connected to the pressure. Similarly, in the case of some fishes, aquatic fishes, you could, if you have read, lateral sense organ, lateral line sense organ, it will be there. Now lateral line sense organ means if you take a fish like this, suppose that this outline of the fish you take. Now here in the body there will be sensory cells. This is what we mean by lateral line sense organs. Particularly deep sea fishes, they have they can feel the pressure wave pressure. They respond to the changing in the the water current will change. So they can feel the change in the water current of the aquatic media they live. Because liquid pressure increases as you go deeper. Okay, according to the depth, HOG, you know that. So liquid pressure increases as you go deeper. Now, so these organs in these fishes help to feel the liquid pressure. At the same time, the pressure due to the change in the movement of the cycle in the water, streaming movement of the. Now, they are on rhonoreceptors. Now, but these are called lateral line sense organ, which respond to the aquatic pressure. Now, here stratosphere is response to the air pressure. Air pressure is what that creates a wavelength wave and the wave motion is felt in this form. And here what happens, the rotation, aquatic, that is semicircular canal, rotation of your body changes the movement of the liquid inside the semicircular canal, that creates it. So statoreceptors comes under the mechanical receptors. Okay, pressure receptors. Now only thing is sound wave, this is the water waves. Now but the lateral and sense organs are present only in the aquatic fishes. Not in the, we don't have the lateral and human beings do not have the lateral and fish. Now these are the three. Now this is a temperature. See, skin, you should know the names for your. Now after this I'll tell you what is needed. Temperature, pain. Now temperature you have got hot and cold. Now this is always they ask, this is one of the multiple choice in the NEET. Now that is the end bulb of cross. Now the end bulb of cross and the Ruffini end. Now the Ruffini end, the end bulb of cross and this one. The Messiness corpuscle will more or less like similar. But only thing is, the end bulb is a bulbous structure. There will be two nerve endings will be there. There's a two nerve endings and they multiply, they branch out inside the structure and the whole thing is enclosed in a bulb-like structure. That's the only difference. Here a single nerve ending will be there, here double nerve ending. They don't round around that thing. They won't wind around the ending. Now they branch out in this. You have got the cytoplasm and the thing. Now this is nothing but bulb is a bulb-like structure. Messiness corpuscle is also round structure. But this one, Ruffini end, is a very funny one. Same thing, you have got the nerve ending. The nerve ending will branch out inside and the outer side, the structure will have an elevated structure like this. This is Ruffini ending. The Ruffini ending, the axon will branch out inside. You have got the cytoplasm and then etc. Whether you understand the structure or not, that is different for your interest. But you should know the name. The pain and pressure, because NEET questions, AMPT questions, they ask you this. The corpuscle or touch corpuscles or stimuli is received by the corpuscles which can touch sensation or warmth. Cold, receptors of cold sensation is cross, receptors of warm sensation is Ruffini. Now this is about 10 to 25 degrees C, not F. And another one is about 30 to 45 degrees C, 20 to 45 degrees C. The temperature beyond that, up to 40 degrees C, you will be able to feel that. However, beyond that, you know very well, boiling point will come. So these are the main receptors of the skin. What you should know mainly, pain, pressure, temperature. In pressure too, light pressure, heavy pressure. Now these names are a must. Messiness corpuscle and Paxinian corpuscle. For pressure. Pain, nerve ending. If you don't know these particular names, at least pain, nerve ending, it ramifies or forms a network on the epidermis. Then cold, you should know the name. End bulb of cross for cold as well as warmth. Ruffini for warmth, cross for the cold. So these are the major pain, pressure, temperature, sensation of the skin. So sensory receptors of the skins are nothing but nerve endings. Nerve endings mean the skin has got two parts, upper epidermis and lower dermis. Dermis has got the connective tissue. In the connective tissue only blood vessels, capillaries, nerves will be there. Epidermis is not supplied to the blood vessels or nerves. There are 4 layers of the epidermis. Epithelium. The outermost layer is a stratified squamous epithelium. First lesson, you should know that. And then you have got next 2 epithelial cells are multiplicated. That is stratum spinosum, stratum granulosum, spinosum, granulosum. And the lowermost layer is important in the epidermis. That is uneven and it has got pigments, Malpighian pigments, which help. It's a single layer which help produce the coloration. Malpighian pigments is responsible for the coloring of the skin. And this skin, this Malpighian pigments helps in the synthesis of vitamin D in the sunlight. So this is the importance of the epidermis. And the dermis has got the connective tissue. It has got the hair follicles. It has got the sweat gland. Sweat gland, everything is placed in the dermis of the skin. And the sweat gland that passes and opens on the upper surface. Root of the hair is in the dermis. And the shaft passes through and the tip only comes above your skin. And the dermis has got the blood vessels as well as it has got sebaceous glands. Now these blood vessels or capillaries will expand or shrink according when the capillaries dilate during the summertime. More sweat formation is there. At that time, filtration rate of the kidney will be lesser. You sweat more in summer, urinate less. But the ulta will be there in the winter season. Urinate more and sweat less. This is due to the alteration, the capillary dilatation of the capillaries in the skin. And the dermis has got the nerve fibers. Now these nerve endings normally passes through the lower part, the papillae, and ramifies and forms a network on the epidermis to receive the sensation. So the skin is the major sense organ. Mechanical stimuli. All the mechanical stimuli can be pain, light pressure, or heavy pressure, or pain, or gravity. Now these are all mechanical stimuli. So the receptors are differentiated: tango receptor, phonoreceptors, and stator receptors. Phono means don't think of the ear, hearing. Phonoreceptors is meant for high pressure. Whether you remember all this already you should know in your inner ear. All these 3 parts you will study in the inner ear. That's not a problem. Now, when you remember this, the high pressure Puccinian corpuscles and Messinus corpuscles are pressure receptors. One is for light pressure, one is for heavy pressure. Remember that. Pain is made by the nerve ending. Though you know all the things, Pakka, what you should know. Pain is by the nerve ending is a receptor. Now, these two names you should know. For heat and for cold. Now, you can frame your NEET question, multiple choice. You can write your own NEET question. So, I can give this, the receptors of the sensory receptors. So many. See, you can say the skin is the major sense organ, the different sensations. One of the different sensation, you can hear mix. That's a dumb question for a lower class kid, I'll ask. Now, skin sensations, when I talk about, a lower class child might not feel as a pain, pressure, temperature. Like that, it won't differentiate. Helps to feel, touch, sense of touch, that's all. But in your question, it can be what? The skin, the major sensory receptors for pain, then hearing, electromagnetic sensation, or light sensation. So pain, when it comes, you will have the main function of the skin. They'll give the different sensations. And you have to pick up that. Sensation means what? I meant pain, sight, hearing. But they won't give in a simple language. Pain, sight, easily you will be able to, in that case, you will be easily able to differentiate. The receptors, the phone, that is auditory receptors. The skin is the main organ which has the receptors to receive pain, audition, sight, or taste. So they will give you the word not so directly. You can give a multiple choice. At the same time, in all these receptors, given the following receptors, which is the one which helps to feel the pain, or which one helps to feel the temperature. Now, which one feels the heavy pressure and light pressure? Messinus corpuscles light pressure, Puccinian corpuscle heavy pressure. Remember that. So they can mix you up. Now they can give you the receptor, which one helps in receiving the sensation, and the receptor's name they can multiply. Particularly, which one of the things help to receive the person touches the water and felt the temperature as 10 degree to temperature cold. So how is he receiving the sensation? With the help of the sensory receptors which receive the sensation of temperature, and they will give the name, mix up all the name, and you have to choose. So these names you should know pakka. Okay, pressure receptors two, Messinus and Puccinian, one for light pressure, another one for heavy pressure. If you, for fun's sake, you can remember that, it's well and good, because the word is given Merkel disc, because the structure will be like a disc form. If you remember the structure, you know that. The basket nerve is always found in the root hair. Now this, if I have this at the paper, I'll say this. Somebody is pulling your hair, a single hair gets caught. You feel an acute pain. Now how this pain sensation is received? This pull of the hair is felt. The receptor which helps you to feel the pull. It is on the hair follicle, surrounding the hair follicle. This is the basket nerve ending, that's what. The basket nerve ending is twisting around the shaft of the hair. The nerve ending in the dermis, the hair, base of the hair is in the dermis. The nerve when comes near the nerve ending, it twists around the shaft of the hair. And that is why it's called basket nerve ending. So you should be able to remember these nerve endings. If you have a chance, you can go through. This is a rough diagram I have given you. You can verify yourself in certain books when you read the receptors pictures. All right. Now this is about the skin. Next you have to go to the skin as a main sense organ. Next we have to go to the skin as a receptor over. Now we will go to the nasal epithelium. Then taste. Now olfactory sensation. Okay. Now skin, all these senses are formed as one, touch. The touch sensation is divided into pain, pressure, temperature, and these are the various nerve endings or these are receptors. Touch is a sensation, pain, pressure, temperature is kind. Now these are the receptors. The receptors are nothing but nerve ending. There in the skin, the nerve endings are there. The nerves are in the lower part of the skin, the dermis. Upper part is epidermis. The ending of the nerve endings or the axonal ending will be on the epidermis. It has to be in the epidermis because in the case of pain, it's only a nerve ending. The nerve ending will branch out in the epidermal cell. But if you take the Puccinian corpuscle, now it won't go inside. It will just end up under the papillae, the Puccinian corpuscle will be there. But They are in contact with the epidermis. Then only you can feel the sensations outside the body. So the first part to come in contact with the epidermis, not the dermis. So from there, another thing. When I say ending, you should not imagine the sensation is forward. What happens? These sensations are carried downward, it goes to the brain. So all these nerve endings are not, the stimuli in that is not the forward stimuli. The backward stimuli towards the CNS. So all these are sensory fibers. So the nerve endings receive the stimuli and then passes it on to the main center. There the stimuli is received and interpreted, analyzed. Sometimes these can be converted to the mechanical stimuli can be converted to the universal conduction, the nerve impulses and electrical stimuli. It's converted into the depolarization occurs. That's what I mean by electrical stimuli. Sound wave or light wave, everything is converted to the magnetic wave or electrical stimuli, then only it passed on to the nerve fiber. So all these sensations, the energy's transformation will be there. Okay, it finally converted in the form of electrical stimuli through the nerve fiber to the central part. Now this is about what you should keep in mind. The stimuli, all the sense organs is not towards the sense organ. It is from the sense organ the stimuli is carried. In the suppose I'm pressure is a stimuli, mechanical stimuli. But the mechanical stimuli, when it is conducted through the thing, transformation of the energy is the mechanical energy converted into the electrical energy. The nerve ending become depolarized. And the nerve stimuli, and that is conducted in the form of electrical stimuli where the brain proper, where it is interpreted, analyzed, interpreted, and back again the sensation comes down as a active mechanical form. Unless movement is there, the response will not be there. When I receive the response, I have to move my hand. That's a mechanical action. So electrical stimuli, when it comes and reaches the effector organ, it is back again, it is converted to mechanical response. Okay. Now this is about the skin. Alright. The next one which we have to go. Nose. Very short is a nose. Nose. This is given in your CBSE textbook, the end of the lesson. Neural control and coordination. Very small paragraph. Other sense organs, and a gist is given. A small account. They are explained eye and ear, and then in between the sense organ, nose alone. They didn't explain the taste, sensation of the taste, because you should study that in the first lesson. That is the human physiology first lesson, digestive system. When it's about the tongue and the papillae and the thing, the different types and how the structure, what is the function of the papillae and what are the sensory cells over there. Alright, now this is nose. Now nose is nothing but the nasal epithelium. Nasal epithelium. See, the nasal epithelium has got three types of cells. Now one is the receptor cell. The cells are like this. This is the nucleus of the world. This is a cell, and there is a basement membrane. Now the nucleus of the cell upper end will end in a knob-like structure, which has got a number of cilia. End in a small knob having a number of cilia around. The body of the cell. They end in a bulb-like structure. You have got a number of cilia. Now this part is jutting out purely diagrammatic. Don't imagine it's all microscopic. Don't imagine it's all poking in your nasal cavity. Then it will be there are hairs, tiny hairs which trap the minute hairs trap the dust particle in the air which you breathe. Now these are microscopic structures on the wall, tiny the projections of the cells, a cilia, number of cilia. This is the knob, cytoplasm of course. The lower end is it's just like a nerve axon like. And in between you have got the huge supporting cells. Now these supporting cells are as huge as touching the basement membrane. Like this is a cell. This is the area where the supporting cells are very large in your nasal epithelium. If you study the structure of retina, you have got giant supporting cells, Muller cells. The full extent of the retina, the cells will be there. You know very well retina has got 10 layers of retina, different layers of the retina. From the upper layer to the lower layer, a huge pillar-like cells are there. Diagrammatically, we say huge, but this huge under the microscope, it will be a microscopic micron level only. But diagrammatically, we should know what are the different types. Now, in the different types of the cells here, now the next to that, these are called supporting cells. It's a large ones, and the supporting cells, below the supporting cell, you have got the basal cells. There are small cells, basal cells. The basement membrane, there are basal cells. In between touching the basal cells, you have got these other basal cells. Now this is the basement membrane. Now these are all cells, basal cells. These are supporting cells. The upper end of the supporting cells have got a number of microvilli. Now it's uneven. It's a number of microvilli like this way, the structure. Now some of these supporting cells will transform into the nasal cells. These are the sensory cells. So nasal epithelium has got three types of cells. One in the basal cell and supporting cell. Now this is a sensory cell. The sensory cell, actually the basal cell sometimes will get modified into the sensory cell. The sensory cell has got an extent, the glob at the end, which has got the cilia. And now this is the entire epithelial lining there. You won't find this so much jutting out in your epithelial lining, nasal epithelium. Now what happens? Nasal epithelial cells respond to chemical stimuli. Chemical stimuli in the sense, the chemicals which you are, the smell, the sense of smell is mainly through the chemical stimuli. Now this is one of the reasons, okay, you know, whenever when you smell any vapor or any perfume, now these air molecules, air only, the chemicals only you are able to sense. Now the another thing is, now this chemical stimuli, that's called accommodation. What is the nasal accommodation? Suppose you go to suppose the cell is receiving the chemical stimuli, it'll get numbed, like, so you can feel the smell for certain this time, a few fractions of the second, and the next minute, if you are keep on inhaling that perfume or the odor, now you won't be able to feel that. That's called nasal accommodation. The nasal accommodation, simple for example, if I tell you, when you take the perfume, when you spray it around your perfume, first time you will find the smell. But when you go out, you won't feel the smell on your body. But the outsiders can feel the smell on your body. Now, if you enter into a place where they sell all this perfume oil, or any place where you are going to get the perfume, what you have to do, the first shot, if you have seen the different people, the perfumed oil they sell in certain areas, they'll allow you here, they'll rub on the hand and then smell it. You keep on, you remain there for 10 minutes, you won't be able to differentiate. Whereas a jasmine smell or rose smell. You know, the different oils are there to prepare the perfume, the basic one. Then they mix it with acetone. That's how your perfumes are prepared. And the essence of these chemicals are sold. Now, if you want to buy a particular smell of a rose or lavender smell or any particular smell, you are very particular, you will get an essential oil. Now that first, within 10 minutes, you should choose. Otherwise, you won't be able to differentiate whether it's a jasmine smell or rose smell. Why? This nasal epithelial cells get accommodated to that smell. So you can't feel that. That's called nasal accommodation. Now that is another reason. People living in a gutter, you go near the gutter, now the people, I wouldn't say living near the gutter, suppose there is some gutter near your place. And first, if you go first enter and you will close it, and after some time you see, you won't feel it, you'll take it for granted. Like, it's not take it for granted, you are not able to differentiate. That sensation is numbed. We won't say numbed, that's called biologically accommodation. Accommodation of the sensation of smell. Now that is peculiarity in the case of the nasal sensation. Stimuli is chemical stimuli, nasal epithelium. There are three different types of cells: sensory cells, supporting cell, basal cells on a basement membrane. Now the supporting cells are as huge as the sensory cell. The end of the sensory cell has got a knob with a number of cilia. The response is chemical response. Now stimuli is chemical stimuli, just like your taste bud. You can't taste the food if that food is not dissolved in the saliva. That chemical only you are able to taste. So chemical stimuli is there. There it is mechanical stimuli, skin and everything. Tongue and nose, you have got the chemical stimuli. Now if you go to the photoreceptors, the stimuli is electrochemical stimuli. In the case of the eye. So here it is a chemical stimuli. The chemical stimuli, these cells are able to receive the chemical stimulated by that, and you are able to, what is the nerve which carries the sensation? Now these nerve ending, there is one cell there may be another cell, say there is another supporting cell over here. The supporting cells have got microvilli. This is a supporting cell. You have got another sensory cell. Purely diagrammatic for you to understand. And the lower end you will have the axon. All these fibers joined together form the olfactory nerve. Olfactory nerve. Olfactory nerve is the first cranial nerve, 1C, a first cranial nerve ending where just beneath the frontal lobe of the cortex. You will see the brain, cortex of the brain, you have the frontal lobe, and beneath that is the olfactory area. Now it ends, the olfactory nerve ends here. Alright, now this is the sensation in the nose. Next you go to the sensation in the tongue. Tongue is a sense organ for taste. Now on the tongue there are small elevations called papillae. Papillae are nothing but just elevations, that's all. Elevations on the tongue. Now if you take the thing, suppose you draw tongue like this way, the tongue has got two different parts. Anterior, this is an anterior region, and the posterior part and anterior part is divided by, this is the posterior part of the tongue. Now the posterior part of the tongue, very good diagram this is, right? The posterior part of the tongue you take, that is separated from the anterior part by a V-shaped groove. This is your digestive system, purely diagrammatic for you to know. The tongue won't be so sharp like that. V-shaped groove, the tongue. Okay. Sorry. One second. It's not coming as I wanted. Okay. Okay. That's what God has given me. I'm sorry. Now, this is the anterior part of the tongue. Now, what is the difference between the anterior and posterior? Posterior part is fully lymphoid tissue. It has no sensory structures. Now, this is known as the lingual tonsil. The tongue is divided into anterior part, front, and the back. Anterior part is sensory. Posterior part is lymphoid. Now, this is called lingual tonsil. Both are separated by a groove. There won't be a depression on a groove. In this, the V-shaped structure, a number of papillae. The papillae are the sense of elevations, small elevations on your tongue. Okay. Now you put out your tongue in front of the mirror. You can't see something poking out. Like that, you will find them uneven. It's not a smooth painted area like. Now there will be small elevations. These elevations are called papillae. Now particularly these elevations, about 8 to 10 elevations, are in a circular form. It's a circle. It's not a cell. It's a circular, a groove and another groove inside. 8 to 10 of them will be arranged in the form of a V. Now what are these papillae called? Circumvallate. They are called the circumvallate papillae or vallate papillae. Circumvallate papillae. These are nothing but elevations. Papillae are elevations. They are not sense organs. Now, because they are in the upper layer of the tongue, the epidermis of the lung, there is some structure there, so the place is not smooth, it is just elevated out. And right in the center of the tongue, you will find a small groove, sulcus terminalis. And on either side of that, you will find little bit smaller but reddish scattered here, reddish in color and scattered here and there. This is called fungiform papillae, fungiform. And all over the tongue, small white structures scattered plenty. This is filiform. Now, what is our famous question? What are the sensory? What are the elevations? They want to ask a sense organ of the tongue, sense organ of taste, because these elevations are not sense, it's a structure. The small elevations, uneven surface on the tongue, these surfaces are known as papillae. Now, what are the different papillae? Add on to that, on the sides of the tongue, there will be small pores. Now in this fold there will be papillae. That's called foliate papillae. The side this is called foliate papillae. Foliate papillae. What are papillae? Elevations of the tongue. And how many types of them? Filiform, fungiform, circumvallate, valate papillae, circumvallate papillae, or foliate papillae. Now, which one of the papillae is numerous? Filiform papillae are numerous. And they are the whitish ones scattered all. How will you differentiate? These are reddish small ones. You can make out when you eat sometimes the sweets or lozenges or toffee, sucking in the tongue. You can make out these things very clearly. And the posterior part of the tongue is a lymphatic tissue that's called lingual tonsil. Now this is the lingual tonsil. Now I hope you could have studied this lingual tonsil when we studied the digestive system, pharynx, tongue. Then posterior to the tongue, you have got the abyssal of the dark area that is called the pharynx, which is a common passage to the air and water. In the pharynx, you have got three different parts. There are two other tonsil tissues in the pharynx. This is lingual tonsil. One in the upper part of the pharynx is called adenoid, and just behind your mouth is the palatine tonsil. Lingual tonsil, adenoid, that's a tonsil, then palatine tonsil. Palatine tonsil is the one you open your mouth wide on either side of the vulva. There will be a mass of the tissue that is called palatine tonsil. This is on the floor of your tongue, back of your tongue, that's a lingual tonsil. Tongue attached to the tongue, so it's called lingual tonsil. But during tonsillectomy, suppose diagrammatically, the mouth is the opening bit here. This on either side, open your mouth wide. On either side, there will be a swelling. This is another insensitive. Okay, just for it done. Suppose mouth, oral cavity. You open your mouth wide in front of it. There will be a small hanging down. This is called vulva. Vulva. Vulva, the folding. Now, on either side of the thing, you will have the tonsils. Now, suppose your tongue is ending here, the posterior part of the tongue also tonsils. So, the opening which is leading from your mouth back to the pharynx, on either side you have got the tonsils, and one is hanging down off on small fold, and the base here, the posterior part of the base, these two won't touch, this is at the side. Now, this is a lingual tonsil. That is a lingual tonsil. Behind that, what will happen? There is a gap through which the food is going to pass. So this is your side of the mouth if you come across. Now you have one is hanging down the vulva. On either side you have the tonsils. Now just behind the mouth. So the lower part is the lingual tonsil. This is called palatine tonsil. Now when this palatine tonsil swells up, what will happen during infection? Suppose you have a throat infection. Sometimes invariably when you eat ice cream, you can make a sore throat. My throat is just choked up like. Now what happens? Now the swelling of the tonsil tissues, you won't be able to swallow even your spit. Now then what is the use of this vulva business? This can move backward. So it can prevent above that what is coming from here, the nasal cavity. So the vulva can move backward and prevent the food from going back to the nose. So it closes the entry, that is the passage which is up. It has to cross and come back. I hope this is digestive system. You should know the structure of pharynx and the lateral section of the thing. What you should know now, the posterior part of the tongue is a lingual tissue. It is not receptor. It is not a sense organ. Anterior part of the tongue only is sensory. What is the sensation? Taste. Now, what is the stimuli? Chemical stimuli. So, what are the receptors? The receptors are not papillae. The uneven surfaces, uneven ridges or small conical structures are called papillae. Now, where are they on the tongue? They are distributed, not smooth entirely. Some are bigger ones at the posterior, separating the posterior and anterior part. That's called valate papillae or circumvallate papillae. Now, why they are called valate papillae? They have a thick round, a groove, a ridge, and the center only the structure will be there. So, in this groove, you have got the cells. Cells, the taste buds are distributed here. So inside the papillae, now suppose you take this as a papillae. Now what is the receptor then? See, if this is an elevation, now this is the lateral section of it. Now this is a elevation, surface of the tongue elevated, and here these are epithelial cells. Okay, fully epithelial cells. The surface of the tongue. You have got the cells are all here. You have a depression here, and there is an opening. This is called the taste pore. Inside this cup-like depression, now inside there is a hole on top. Now this is actually taste bud. Now this is the surface of the tongue lined with all the epithelial cells. The cells are there. Cells are all there. Okay. Now in the taste bud, you have an opening on top. This is called taste pore or a pit. Now inside you have got two types of cells. One is a sensory cell that's called gustatory cell. It's a cell like a neuron. It has got one end will have a cilia which will be poking through that. Another end you have got the lower end. Now this end joins with the there will be a nerve supply there. It synapses with the axon of another one that will go to the cell, and these fibers form the nerve. Continuous the sensation is conducted like this. Now in between, just you saw the nasal epithelium as supporting cell. Here also you have got by the side you have got a number of supporting cells. Packing it up. Supporting cells are just packing it up. I am just showing you one cell only. Now similarly within that 1, 3, 4 cells will be the sensory cells, and through the opening the cilia will be coming out. They are all packed. These are called supporting cells. So sensory cell, supporting cell. The lower end of the sensory cell will anastom or synapse. The axon of the nerve, all these nerve endings, they will form the nerve endings, and that gives to the sensation to the brain. This is the taste nerve. That is it. These fibers form the glossopharyngeal nerve. The sensory fibers ascend to the brain via the ninth nerve. The ninth nerve, cranial nerve, is known as the glossopharyngeal nerve. So taste sensation is conducted to the brain through the ninth nerve. Tongue muscles are supplied by the 12th nerve. Muscles are motor, so 12th nerve is hypoglossal, that supply the muscles of the tongue. Glossopharyngeal is the 9th nerve, that's a sensory nerve for the tongue. Tongue has got 2 nerves supplying it. Do you follow that? So this is what actually mean by taste bud. So which actually the sense organ? The taste bud. Where is the taste bud? Taste bud is nothing but an assemblage of, you have a sensory cell with the cilia, the nucleus, and the lower end it comes axon, and you have got the supporting cells in the thing. Now these are just like a nasal epithelium. Supporting cell, you can have another gustatory cell, and these fibers, these gustatory cells will have the cilia which will come out. The whole thing is inside the papillae. Now what is papillae? Papillae is nothing but elevations on the, now the whole thing is the elevation on the tongue. These are lined by this is the tongue surface. Now these are all the cells of the tongue. So this whole elevation only is known as papillae. This whole elevation is called papillae. Actual sense organ is what? Gustatory cells. Gustatory cells or sensory cells. Now these are all in between. You have got supporting cells. Now there is a hole or a pore. Through the opening, now the cilia of the cells come out, and this cilia receives the chemical stimuli. A food matter dissolved in the saliva. If there is no saliva, it doesn't dissolve. You cannot feel the sensation because it's a chemical stimulation. And the nerve fiber from here, the lower part of the cell, the fiber and the axon, they form the axons, and the fibers of these cells join together, forming via the ninth nerve, it reaches the brain. So there are 4 types of papillae. So inside each papillae there will be taste bud. Now what is the difference in the circumferential papillae? There is a groove. On the sides of the groove, the taste buds are scattered. Now in this all like this cup-like structures, filiform and fungiform. Now so papillae is different, taste bud is different. Taste bud is within the papillae. Papillae are nothing but elevations on that. So where I can ask, you can frame your own multiple choice question in this way. So which part of the tongue is the receptor? Or the type tonsils in the tongue is known as the lingual tonsil. Or lingual tonsil belongs to anterior part of the tongue or posterior part of the tongue, or you can say on the pharyngeal region or in the nasopharynx. You should know the difference of the nasopharynx, oropharynx and laryngopharynx. Now the lymphatic tissue in the nasopharynx is adenoid. Oropharynx is palatine tonsil, and either side of the buccal cavity, the opening. And the lower part of the opening is a lingual tonsil. So which tonsil is removed during the tonsillectomy? Palatine tonsils are removed. Now when does this swell up? When there is an infection, this lymphatic tissue swells up and blocks the whole thing. When you have a severe cold, you can feel it. When you have a throat pain, you can feel it. You can't even swallow your spit. The best first aid treatment is what? Just gargle your mouth with a concentrated salt solution. Salt as much as you can. Well, take it nicely, you gargle. Hold the salt on the throat. That's the upper part of your pharynx. Just behind it. Now that is the best first aid for this tonsil, swelling of the tonsils. It indicates in olden days, that's a few days when we are small, they used to remove this tonsil. They'll operate it. When they are small children, will struggle a lot. But recently, they don't advise that because very well tonsil lymphatic tissue gives you resistance. So the natural resistance is one of the lymphatic tissues providing the natural barrier. Very well secondary lymphoid organ and primary lymphoid organ, lymph tissues. So the lymph tissues is the one which protects you, gives you immunity, lymphocytes. So nowadays they don't advise you to remove the palatine tonsils. You follow? Now that is about the pharyngeal region. But when you come to a sensation, which is a sense organs or sensory receptors of your tongue, don't write papillae, gustatory cells or taste bud. Now this whole thing is known as a taste bud. Now taste bud has got two types of cells, gustatory cells, receptor cells and the supporting cells. Receptor cells will have a cilia which will come out through the papillae, and the lower end got the nerve fiber. Now the lower end got the fiber which will form along with the forms a sensory nerve fiber which conducts the stimuli upward via ninth cranial nerve, where to the central area, the cerebral cortex. If you take the cerebral cortex, where is the cerebral cortex diagram? Gustatory area. If you take the temporal region, there will be a fissure, longitudinal fissure, central fissure, groove, longitudinal groove separating the upper part from the temporal lobe. In that part, it will be given gustatory area. That is the area where the sensation is received. The lower part of the sensory area, end of the sensory area. They say, I hope you should know the diagram. See, whether you approach for NEET or your public exam, you can't study biology in the air. This is not like writing the formula or working on the problem. First, before you learn the part, you should draw the diagram. Look at the diagram, even though you don't understand, either it's a curve or straight line, whatever, draw the diagram and label the diagram. Then you go for description. Never start learning the biology straight away the answer. If you don't know the diagram, you can never master your biology. Any diagram, you should be able to draw your own diagram. So familiarize yourself before you want to thorough. Familiarize yourself with the figure. Rough figure is enough. You need not be able to draw exactly as in, but if you draw the diagram, you see the actual structures in your body. It won't like the diagrammatic. These are all diagrammatic explanations, not the real tissue. Of course, you could have seen tongue. All right, but these things are all further analysis only. So this is about the now. Which area? Now we are in the sensation. Now what are the different taste sensations? Bitterness, sweetness, sour, and spicy. Now when you take about the sweetness, now if you take the tongue, the posterior part, this is about all the papillae. If you take a tongue region, if you take a tongue as it is, now if you take the tongue, the posterior part, the groove is there. Now this is the area where you will feel the bitterness. Now the tip of the tongue is sweetness. Okay, the edge of the tongue, the lateral edge of the tongue is a sour. It will be given lateral edge. If the tongue is a sweetness, this is sour. Now if you take a tongue, it's not a flat paper. You can pull out the tongue and feel and see. It will not be. It will be a thick space, a thick one. So if there is a thickness, there will be lateral side. There will be tissue at the side. So this flat one, there will be edge here or edge here. This is the lower edge. Now this is the lower margin, we can say. Yeah, tongue is not a flat thin paper like this way. It's a little bit thick muscular structure. So when you have a side wall, you won't even say side wall, side of the tongue. This is the lower margin. This is the margin. This is the edge. So the edge of the papillae, which helps you to see another sensation, that is sour. The one up is the salty. So the sweetness, tip, Bitterness, sour will be the tip as well as by the side, edge, lower edge. Then salty is a little bit above here, this portion. So when you say margin and the lateral, it will be described in your book, it's a lower edge and the margin. That is, imagine the muscle tissue is so thick, the slight elevation, a ridge will be there, below this is the edge. The edge will be along the papillae. Now that is the edge one which is here, that will come to the tip also. So suppose the bitterness takes a little bit longer time, a fraction of the second you feel this slower than the sweetness. So that is another advantage, because most of the plant products or alkaloids are bitter in taste. And now the tip of the tongue is only sweetness, and the sourness will come. That is why when you eat the sour things, it will be totally sour. There will be a tinge of sweetness also will be there. Any sour fruits which is, you eat the gooseberry or tamarind, anything, you will find a sense of sweetness as well as a sour taste. That is mainly the sourness is all along the edge of the tongue as well as in the tip. Now this much is your sweetness. Now if you eat the food, the sweetness as bitterness, first you will feel the sweetness, then only bitter. Now this is for the lateral edge, lateral side of the, on the sides will be the salty. Salt at the side. Now this is the lower edge. Lower edge. Edge will be the sour taste. Now this is sweetness, sweetness, and this is bitterness. So these are the major sensations of your tongue. Okay, now we go to the first one. So we have skin, nose, tongue, we have to go to eye. Now you know we are very familiar with your eye structure of the eye. The eyes are the vision, you know very well. Now these okay, now the sight, main organ of the sight is the eye, and which organ acts like a third eye, pineal gland. If you have studied our endocrine system, pineal gland acts like a third eye in the case of that, it can receive certain sensation. Now sensation stimuli are photochemical. What is light waves? You know, light waves are visible range and invisible range. I hope you know the light rays section. Visible spectrum extends from 450 nm to 7 to 750 nm. Our visible range extends from 380 nm to 760 nm. That much of range, your eyes, visible range of your sunlight. Sunlight, as it is, has got the visible range, and the high. These are, you know very well, sun solar energy. So the solar energy is in a visible range, is the one we call that as visible light. Now there are energy above the visible light and below the range. That is the high energy rays below your visible range of 350, 450 if you take, below that 400 that way, below that, and above that. The rays which are above is known as the infra, and which is below the ultra. So if you go below the sunlight, if you take the range of the sunlight, if you have the sunlight range, this is a visible spectrum. This much is visible spectrum, 400 to 750 you take. You know very well the 7 lights are there. In that, Vijaya, violet, indigo and blue will be merged with one another. So you won't exactly see one violet and indigo and blue like that way. So this, but all these have got a minimum difference of 50-50 nm. 400 to 450, 450, 500, 500 to 550 it will become a green. Like, you know, the range of the green light. What do you mean by that? The wavelength of these rays. Each ray, each color is a ray, the light rays. The rays has got different wavelength. Now, what is the wavelength? The distance between the crust or a trough. You know the transverse wavelength. The longitudinal wave, it will be compression between consecutive compressions or rarefactions. So if you take up the wavelength of a visible spectrum, it is 400 to 750. In that, the green light is 500 to 550 it will come range. So we are starting with this V and this is the end R. So in between, say 500 to 550, 500 to 550 nm, you will have the green. That is, this ray will have the wavelength of this nm. Now the ray here, it will have the wavelength of this nm. This is the ray. That's the meaning of it. Okay. Now there are rays below this and above that. Now this is infrared. Infrared and heat rays. The high energy rays above 750, first comes what? Infrared, and leads to microwaves, that leads to radio waves. Now below what you have? UV, below that is gamma, and below that is, what is below you have? UV rays, gamma rays and X rays. That is below. But the infrared ray, what is the effect of infrared? Heat effect. Completely it produces a heat effect. Now this one, you cannot feel that, but it can penetrate through the tissue. Now we are only in this visible range. So visible range is between this 400 to 750 mm. So our visible capacity is between 380 nm to 760 nm. That is, you remember 380, double 380, 760. Remember like that. Audible range is 20,000 millihertz. Similarly, your range for your visible range of a light ray, our eyesight is 380 to 760 nm. It's a vision. And the vision also you have got different. Now, some animals can see in the daytime, not see in the nighttime. You have got the receptor cells, photoreceptor cells, rods and cones, you know that. Now, these are the sensory cells. Now, one cell helps in the vision in the daylight, another cell helps in the vision in the dim light. That is, if you take cones, cones help the vision in the bright light and color vision, both. Cones, but the rods help in the night vision, scotopic vision and photopic means daylight, scotopic means twilight. So the cones help you to see in the, rods help you to see in the twilight and dim light. The other one is the bright light as well as the color, two functions. For the cones, one function for the rod. So if that particular, some animals which are blind in the night like a bats, that means what? They do not have in their eye, cones are absent totally. So they can only see in the night vision and day vision. So those animals are nocturnal and diurnal. Nocturnal vision means they cannot see in the, some organisms can see only in the night but not in the day. Nocturnal. Now now the vision depends on the two main sensory cells, rods and cones. These rods and cone cells is the longest cell, the largest cell in your body. If you have seen the electron microscopic structure, if you study the rods and cone, you can appreciate the difference between animal cell, normal single animal cells which are growing, and the rods and cones. It's a very lengthy cell and long one. The whole cell itself would be divided into four parts. Upper segment, lower middle segment, then you have a end tendon and the middle part, the nuclear part, and that ends in the receptor end. The receptor end will be different between the rods and cones, and in the upper segment we see the whole body. The rods and cones are very lengthy cell. Now we will see when it comes to the eye. So this is about the light vision. The visible range, what is our capacity, and how do we receive? Mainly receptors are sensory cells, and the light sensory cells are rods and cones. The organ first is our eye. So structure of the eye. The structure of the eye you know very well. You know very well the structure. Eyes are placed in the socket in the skull. 1 second. The structure of the eye. The eye is placed within the bony socket, cavity in the skull, And the cavity itself is not a single hole in a bone. If we study how the cavity is formed, the number of bones, upper part you have got the frontal and the lacrimal bone, the side lower part is the maxillary bone. So all these bones which are surrounding the socket area, the cavity is formed by these bones. And the nasal bone this side, a small bone is the lacrimal bone. So now the eye socket in the skull, is made up of the different bones, surrounding bones form a cavity. Now the posterior part of the cavity, now there will be an exit. That is, there will be a space through which the nerve, which is the main thick rope-like nerve that's called the optic nerve. Now this optic nerve moves, that is, comes out from the eye. The eye is the part which we see in front is only the little portion of the eye. The entire globular part of the eye is within the socket. But when it is in the bony socket, eye is not fixed or fused with the socket. It's a free ball inside the cavity. Like, it's free, it doesn't fuse with it at all. So behind is a rope coming out. So if anything happens to the rope, if I cut the rope, I can push the eye outside the eye socket. Any violent bang from the posterior part of the head eyeball can pop out. If it gets cut in the optic nerve, the whole eye can come out. The pressure is more. It's not a long rope, your optic nerve, but it's a thick nerve which is emerging from the posterior. It's a globe, ball-like structure. The posterior part of the a rope-like thing will come. That's the optic nerve, which comes through the opening in there is a space in the posterior part of the socket of the eye. Now, where does this optic nerve enter? Where it is present in the brain? Hypothalamus, pituitary gland. In front of the pituitary gland, the optic nerve is there. The optic nerve crosses from the left to right, right to left. That's an optic chiasma. That is the left part of your eye. Left eye nerve reaches the right part of the thing, and the right eye left part of the brain proper. So the optic nerve is a rope which suspends the ball-like structure within the cavity. So the cavity, the eyeball, has got a posterior part which is within the bony case. The anterior part, what we see slightly to the very little space. And that anterior part is protected by the eyelid and eyelashes. Eyelashes are like a curtain protecting from the dust particles. And the eyelid, you know very well, and the lateral part, the left corner of the eye, the lateral end of the eye, you have got the lacrimal gland, which poses a lacrimal secretion or the tear gland. And apart from this, there is a thin liquid of water will be there on the surface of the eye. Now, before you go to that, what it is made up of, you should know that eye, even you studied, you are making a ball with 3 different color paper, you take it. Now, outermost, first you just take a roll of paper, cover it first with the cover, 1 color paper. Now, this color paper, you are not completely, you must have a 3-dimensional figure. Say, suppose I take a globe, globular structure. It has got like this. I have rolled up a paper. Now, completely, I have covered with the 1 paper, first color. Completely, it is covered with the first paper throughout. From here to here, I covered the paper. Okay. Now, the entire, I have rolled something round inside. I have got, maybe I have taken a paper roll, nicely make a paper and take 3 color paper. Outermost color paper, this is the outermost, completely covered and slightly jutting out the ball. It's not a spherical structure. Okay, this whole thing is covered completely. Now I am taking the paper a little bit forward in the front like this and covering it up. Then second, what I do, I remove the paper. So the first paper is removed. Now I have removed the whole thing, ripped out here. So only the edge I can see there like this. Alright. Now after I remove that, there is another second cover. Now the second cover, what happened? This is the cover, okay, outermost cover. I have ripped out in the middle part, I removed. So only the edge of the skin is left out. Edge of the first cover is left out here. Now the second cover, what I am taking. Now the second one. Now the second cover. On the next one, I am covering the second paper, the second covering. This is completely the second cover. Okay. Now this second cover, what I do, I am not covering it to the front part. Okay. Now this second cover, the full paper, Now the edge of the cover, this is like this. It's a spherical structure throughout, front as well as back and covering. All right. Now the one end of the cover, I have the paper, I have made a little bit ridge like this. Like this, okay? Ridges like uneven. Then after folding the paper a little bit, I am taking it like a smooth lining. Smooth, very smooth outline. But this smooth outline, I am leaving it like this and leaving this whole thing is the thing, and there will be the fully it is I am covering it up and down like this way. So when I am bringing it, there will be a space I left out. So the space I left out in the center here. This is the second cover. The second cover completely covered inside posterior chamber. I am putting a second paper, then make the paper a little bit thicker at one end. Then from there I take the paper smooth like this. But the anterior part I am not joining, there is a gap I left out here. You understand that? So now this is second cover. So if I remove the second cover, what level? Suppose I am removing the second cover. I have got already the first one. Now which is there remaining already and going up like that and coming down. So this is the outermost one which is already taken and going up like this. This outermost peel. Then the second one also I am peeling off. So the second one I am peeling off, how the outline will be there. Now the second one will have the same thing. This much is the second one. Now the second one, the second layer. Okay, the second layer. What I do here, I made it as a thick, no? So that end will appear a little bit thicker. This will be thicker. This is the second part. It will be thicker. And then there is a smooth part, that will be hanging down like this. You follow? So this is the smooth part of the second layer. Now this is a little bit uneven part of the second layer. Now this is on the posterior side. So the second cover I ripped out. So that the second cover posterior I have peeled off like that. So this are the edges, this thick areas are the edges like this, and the one which is smooth, it's sweeping out is like this way, hanging down. And the smooth lining is not joining, there is a space over here. Do you understand that? This is the second layer. Then we'll go to the third cover. Now, so first one, second one here. Now third color paper. Now third color paper, what I'm doing, I'm just covering up till this. So the third color. What to take? Okay. Now the third lining covering fully covered like this is the third paper cover fully. Okay. Now this is the third paper which I am covering the globe, the eyeball. Okay. When it reaches here, that is before this thick area, below this. Now thick area, what I do, the third cover, I am just folding it like this way, ending it up. That much only the third paper. And when it beyond that, now the entire paper, the third color paper, I have rolled it up like that. A thin lining I am going up to this. The third paper, a thin lining going up to this, even up to this it ends. Okay. Now I remove the third paper. How will it look like? So if I remove the third paper, right? Now first. Now the first lining is this outer one. Okay, the outermost, the first cover, that is this one, completely covered. And the anteriorly it goes as the bulging out area. Now purely diagrammatic for you to understand only. Now this is now this is the first one. I have removed, so anterior pushes out. What is the second one? The second layer. What is the second layer now? Now this is the second layer. The second layer is this. It's not a single line because it's a layer. You can make out the thickness. This is the outermost layer. The first cover which I peeled off. The first cover which is going on up to this, it will be thinner anteriorly. Anteriorly it becomes thinner like this. Okay. It's not as thick as the posterior. Posterior means back part. This is the outermost first covering. First color paper which I ripped out. I removed here and showed you what at the side. Alright. Now the second one. The second one, what you have to do? I am taking the second one and it is uneven. Here it becomes uneven. Then it forms a loop. It hangs down as a loop. It ends like a loop. It's attached like this way. Okay, this is second layer. So similarly here, you have a second layer. It forms a structure. Then it forms a fold. Joins along that. So fold. Now this is the anterior part is folded. This is the second layer. Alright. Now this is completely the second layer. Now what is the third one? The third layer. And now I have to see the third covering. Now third covering is, it goes only now up to this. This is the third covering. The third covering. Third covering. It ends up like this way. It ends up in a line. Okay. Now that it is just ending up like that. After that, how it goes? It goes as a thin lining over the structure and fuses like that. You follow? Now this is actually the three layers of your eyeball. The eyeball contains three layers. When you draw diagrammatically, you should imagine the whole globe is covered with three different color papers. What they are showing is, they are ripping out one by one and showing the line. So what will be the first one in this? So in this, what happens? Another structure. Now here, in the eyeball, now this portion is this is called the iris and this is ciliary muscle. You have a lens kept here and lens is attached to the ciliary muscle by the ligament. So the lens is right, there is an opening here. Now this gap. Now this is a lens kept here and it is attached onto those projections. Now the gap in this. Now this is the hanging down, there is a space. Now there is a gap. So these are the different layers of the eye. First layer, second layer, third layer. First layer goes completely, surrounds at the back as well as in the front. Now second layer, it forms a uneven structure, the thickening here, that is what I showed you, the thickening here, then goes smooth up. So the thickened area, side view, it will be like this. So the thickened area will form a structure like this. And it forms a curtain hanging down. It's not closing completely, there is a gap. This one continuous goes up. So the second layer, it forms a irregular structure over here and then hangs like a curtain in front. This is the second layer. Alright. Now the third layer has, which is this one, that third layer does not extend even up till then. The first layer goes completely front and back. Second layer forms halfway through. The third layer does not even reach that much. It ends in the thing and forms a thin lining layer there. And this projecting area, there is a ligament which holds the lens over there. Now this is actually the different parts of your eye. Now see, if you have to continue the light. This part. closer. Okay. Now the posterior part of the this innermost layer has got two parts. That is the posteriorly it ends the thickness here. Now this is the sensory part. Now if you label the parts, you will know the different layers. It ends here itself. This is called ora serrata. Now the whole thing you have got the through the space the fibers from here comes out. That is the posterior innermost layer has got the sensory cells. All the axons of the sensory cells come out when they come out it's an optic nerve, and at the same time the blood vessels enter into it. So through the opening, the posterior part, this is eyeball. Now what are the different parts you have to label now? See this one. This is anterior part, that's a posterior part. Apart from these three coverings of the eyeball, the anterior, this is front and this is the back. Anterior means front, posterior is back. There's eyeball inside the skull. This is the one what you're seeing outside. So apart from this, now the anterior part, which is outside, this has a thin another covering that comes out and reflects over the eyelid. Trapex, this is your eyelid here. It goes and that part which you can see comes out. This is the conjunctiva. Now what are the different parts? This is the first layer. Posterior part is known as the choroid layer. Choroid. The choroid goes anteriorly. This is known as the cornea. Cornea or the white of the eye. Now the second one is, I'm sorry. Now the first one is a scleroid. Scleroid. Anteriorly it's known as a cornea. The second layer is the choroid. Choroid, the difference is, this is nothing but a connective serous layer. But the choroid layer is a pigmented, completely pigmented cells. So the pigmented cell keeps the posterior chamber very dark so that it prevents the refraction of the light. So the choroid layer is the second layer. When it comes anteriorly in the front part, it doesn't go up like the first layer. Now here it forms a structure called ciliary body. Ciliary body. And it falls into a small curtain. That is, now this is the ciliary body. Okay. Now this part is the ciliary body. And the whole smooth area is the iris. So when you draw in the diagram, lateral section, I can show you like a loop hanging down. But actually, the whole iris, now what will happen in reality? This iris has got the pigment color. Now that gives a color for your eye. So you find the black of your eye, the round of the eye, which has got, that is the iris. So the iris is a curtain. But diagrammatically, when you draw, you should not imagine it's hanging down. The whole paper is like that. That is, if all my finger is iris, it's coming from back, it's like this, ending up completely. And there is a space inside. So when I cut a section, I can see only the edges alone. I'm showing like this in the diagram section of the eye. So this is the iris part. This is the ciliary body. Now this is the posterior. The whole thing is a choroid layer. So the iris and choroid layer, and at the end of the ciliary body, you have got the lens, which is supported by the suspensory ligament. Now, so this is the ciliary body. Now this is iris. And what is this gap? This gap is known as the pupil. So walk through the gap, you can see at the back the lens. So only the part through the gap, it's like a pinhole camera. That is called the iris. You have got the pupil. Pupil is known as the apple of your eye. Through the hole, what you see, transparent area, what you see behind, that is the lens. So how the lens is kept over there? On either side of the lens, you have got the ligament. Now, on either side of the thing, you have got the ligament. That is, this is a ligament which attaches holds the lens in position. Now, this is the ligament. Do you follow? So, the innermost part, this is the retina. Now this is a sensory area which has got a photoreceptor cells of rods and bones, and the cells are separate, distributed throughout. And the outer surface rod, there are two types rods and bones, and the rods are more toward the outer surface than toward the inner surface. Okay, now these rods and bones, this layer it does not go with the front part of your eye. It stops behind the lens. The membrane stops like that. Now this edge of the retina, this innermost part is the retina. Now this retina edges at this is the sensory part of the retina. Now this is a sensory part of the retina. It ends anteriorly. It ends at the edge known as the ora serrata. And beyond that, the entire thick layer won't follow. One thin layer of the retina will line over the ciliary body and the inner surface of the iris. So the retina, you have got three parts. The posterior part is the sensory retina, and this is called ciliary retina. This is called iridial retina. That is only posterior part. They have the sensation. They have sensory receptors. They can receive the photoreception, light waves. They can respond to the light waves. Now there are two types of cells, rods and cones. And the sensory part of the retina stops. That is, the whole sensory part stops like a ora serrata. Beyond that, it runs in a thin layer lining the ciliary body as well as iris. So the retina has got sensory retina, end the ora serrata, continues form a thin lining layer over the ciliary body, ciliary retina, and iridial retina. Yes, ma'am. So the lens is suspended by the. Now there is one front part, one is the posterior part. Now you have got a chamber. Now there is a chamber here. Now you have got a posteriorly on chamber. Now this is anteriorly on chamber. There is a gap between the lens and the irises. So the whole portion is called anterior chamber. This as well as this. Now this whole thing is in the posterior chamber. The anterior chamber is filled with aqueous humor. This is called vitreous humor. It's a liquid, it's a colloidal substance. Now the anterior chamber, they have got a front larger area and a small gap between the iris and the lens and the ligament. Everything comes under the anterior chamber. And it is filled with a liquid aqueous humor which is secreted by the ciliary body. So now then anteriorly protecting a thin layer or the one covering the anterior part is called conjunctiva. Conjunctiva, this is the white of your eye. Now the central part, the black of your eye is iris, and the center of the black curtain is the pupil, and the conjunctiva comes and reflects over that. Okay, now please try to draw the figure and label the different parts, and the retina, what are the different parts of retina, what are the different parts of the second layer, what are the different parts of the first layer. We'll proceed in the next class. Thank you.
RGR Academy is the NEET class. NEET